welcome to the HIV Cure podcast. My name is Sakabo Marumo, and we're just here to bring to you HIV news. We're just going to break HIV from apart to explain to you in simple terms for you to understand, and also just to get the community to understand the dynamics of HIV cure and also have their voices, to have them talking. I think now more than ever, we do need an HIV cure around the world and in Africa, since, we are the most hard hit by this epidemic. So today I have another exciting guest with me. Hi, Adam.
Hi, Sagabo. Thank you for having me.
Thank you for making the time. And today we're going to be talking about the types of HIV cures that are there, and we're specifically going to look at functional cure versus sterilizing cure. But before we look into that, please just introduce yourself and let us in on what you do and why you are my guest today.
Yes, hi. So I'm Adam. I am the research program manager and also statistician for the research enterprise to advance a cure for HIV. So we are an international group of scientists committed towards to advancing a cure for HIV, based out of New York, but again, we're international, and we're funded by the U.S. National Institutes of Health through their flagship program called the Martin Delaney Collaboratory Program for HIV Cure Science. And I have been working now in the HIV cure field, researching HIV cure strategies for about 8 years now. So I do broadly HIV cure and also what's called HIV persistence research. And so, our research is focused on trying to understand how and why HIV persists despite effective antiretroviral therapy. And we also investigate cure strategies from what's called basic science, really lab-based research, all the way through to clinical trials.
Wow, that's a lot of work. And
it's a time, yes.
And yeah, it's really exciting work to hear you speak about it. And of course, we're going to learn more as time goes on. And then, as the season ends, we're not going to just put everything together to say, oh, so this is exactly what he does. We've also just learned about the HIV language guide, that it was made in 2020, and it was and then it was shared with everyone like in 2024, the most updated version of it. And in that language guide, it just talks about us having to change some of the terms that we used to use. We know that HIV has been there for over 40 years. So some of the language that was used, maybe let's say 20 years ago, now we have to change it, or even just recently, some of the language and terms have changed. So before we dive more into today's topic, please explain to us what the HIV language guide is.
Yeah, so the language guide is really about centering a person-first language, and so respecting the individual, the person living with HIV. It builds upon what's called the Denver Principles from the 1980s, and it's person-centered language to destigmatize HIV. So you mentioned this term sterilizing cure. This is a term that people still use. You may also see this term in the published literature, the scientific literature. It is an older term, and the community preferred language for this is now what could be defined as classical definition of cure, or eradication or eradicating cure. And so the concept here is elimination of all HIV-infected cells in the body to prevent viral rebound if a person living with HIV discontinues their antiretroviral medication. And then you also mentioned a term called functional cure. The community preferred language for this is viral control off of therapy. And so here the goal is to allow a person living with HIV, their immune system or some other type of intervention to control viral loads to undetectable or to very low levels without the need for antiretroviral medication, thus promoting the health of the person living with HIV and preventing the risk of transmission. So in this case, in the concept of functional cure or viral control off of therapy, which is the term I'll be using going forward, the person is still living with HIV, but it is being controlled to low levels.
Okay, so it's just basically the use of person-centered language. And then last year also, we did see at the AIDS 2024 that IAS, also the International AIDS Society, also stresses the use of this person-centered language. We also saw that last year's theme was put people first, which is just basically about putting people first, and making sure that when we're speaking and writing on anything that has to do with HIV, it's always we use person-centered language. So for today's episode, we are going to do that, but like you did say that some old literature does always refer to the old language. So we're going to just go back and forth within with the terms that we're going to be using. So it's not wrong, but we're just going to keep sometimes referring back to functional cure and sterilizing cure just so everyone understands, but we also refer to the new terms that we're supposed to be using that are now person-centered. Is that fine?
Yep, that's very fair.
All right. So yes, functional cure now is called viral control off of therapy, and then sterilizing cure is now termed as complete HIV remission or the eradication cure that you did explain on. So now that we're speaking of these big terms already, please just explain to us what the differences are. What's the difference between this sterilizing cure and functional cure?
Yeah, so, as I noted, sterilizing cure or eradication is the concept of eliminating all HIV-infected cells in the body. So in practice, this is likely very difficult to achieve. It's also very difficult to measure in a person living with HIV because we can't sample every tissue site where HIV hides out. And so it's very difficult to demonstrate that eradication or sterilizing cure has been achieved. And then functional cure is the concept that someone is still living with the virus. There are still HIV-infected cells, but they're able to control the virus to lower levels in their blood without the need for antiretroviral medication. So two different concepts, and we'll talk a little bit more later about different cure strategies that try to achieve these outcomes and these goals.
All right, so now we know the difference. So then why is it so difficult to then completely remove the HIV from a person's body, even though, even with this powerful HIV treatment, the ARVs that we have, why is it difficult for us to just remove the virus from the body?
Yeah, so that's a very important question. We know that antiretroviral medication suppresses the virus from replicating, and modern antiretroviral medicines are very effective at that, and they're also very safe. But the challenge with removing HIV lies within its biology. So HIV is what's called a retrovirus. This means that it's a virus that converts its RNA genome into DNA when it infects a cell. And so DNA is what our genetic material is made of, our human genetic material. So when HIV infects a cell, it permanently inserts, or what's called integrates, its new DNA genome into the human DNA of the cell that it infects. So in this way, once HIV infects a cell, that cell has to be killed, such as by the immune system, in order to remove HIV from the body. And so cells that persist and that continue living with HIV inserted into that cell, those cells form what we call the HIV reservoir, which I'll talk about more in a moment. So as a side note, some groups are working to remove HIV DNA from infected cells, such as using CRISPR genetic engineering, but this technology is still in very early experimental development. So the predominant cell type that HIV infects is called the CD4 T cell, which is an important type of immune cell in the body, and this is why CD4 counts are monitored. So CD4 T cells can be located in many different areas or tissue sites throughout the body, including tissue sites that exclude other killer immune cells, thus making it difficult for the immune system to remove all HIV-infected cells. Also, there are other types of cells which HIV can infect, some cells called microglia or astrocytes in the brain, tissue-resident macrophages. And so collectively, along with CD4 T cells, these infected cells that persist despite antiretroviral therapy form what we call the HIV reservoir. And then additionally, a major challenge with HIV is that it can go dormant or what we call latent. And so here, cells are infected with HIV, but they are not producing new viruses. And so there's no way here for the immune system to recognize these cells as infected. There are some drugs, a class of drugs called latency reversing agents that have been tested to try to reactivate or reawaken latent or dormant infected cells. But currently these drugs are also still in early experimental development, and they're not able to reactivate every latently infected cell in the body. So the persistence of these latently infected cells is a major challenge with eliminating HIV and curing HIV. And so the HIV cure field is really focused on trying to understand this pool of dormant or latent infected cells called the reservoir. So to sum all that up, antiretroviral therapy, HIV medication, stops the virus from replicating, meaning it prevents new cells from becoming infected, but it does not eliminate this HIV reservoir. And so a person living with HIV must continue taking antiretroviral medication to prevent the virus from rebounding to detectable levels in their blood.
Okay, and then now I want to understand. So when someone achieves this functional cure or the viral control off of therapy, do they still have this HIV in their body, and then can they also transmit it to someone else?
That's right. So viral control of therapy, by definition, means that the person is still living with HIV, but their immune system is controlling viral replication, very similar to how antiretroviral therapy works. So the goal for viral control off of therapy is to allow the person to maintain undetectable or at least very low viral loads. So it's not currently known, however, if this is achievable in all people living with HIV and how long this viral control can be maintained. So the HIV cure field is still very actively testing strategies to achieve viral control in clinical trials. Currently, most clinical trials are very early phase clinical trials. There's also a lot of preclinical testing in animal models and in vitro testing in laboratories. So I will say there are currently a few cases, exciting cases, of viral control off of therapy from a few different clinical trials that have been conducted around the world. And we are trying, the HIV cure field is trying to better understand those specific cases and how those individuals are controlling viral loads off of therapy, whereas the majority of participants in these clinical trials did not go on to control viral loads off of therapy. But to get back to your question about transmissibility, this will depend upon the person's viral load. So conservatively, a viral load of about less than 200 copies per milliliter of blood plasma is thought to substantially protect from risk of onward transmission, but it will depend upon what we're able to achieve in different interventions and what the person's viral load is. So the goal is to maintain very low viral loads, but again, the field is still what I say in its infancy, and we're still testing different strategies to achieve viral control, and it's not known what strategies are going to work in the majority of people living with HIV.
Okay, and then you mentioned some exciting cases that have happened or that are currently ongoing. Can you please share with us some of those examples of people who are now functionally cured around the world?
Yeah, so I'll start with eradicating cure, and these may be the cases that people are familiar with through bone marrow stem cell transplant. And so to date, there appear to be 10 cases of what could be called eradication. So in each of these cases, the person living with HIV received a hematopoietic or bone marrow stem cell transplant to treat an underlying cancer. So they had a type of hematologic malignancy that required this bone marrow stem cell transplant for treatment. And then as a result of the transplant procedure, these individuals appear to be cured of HIV. So I say appear to be cured because it's impossible to sample and measure every tissue and every cell in the body to determine if HIV is truly eradicated. But these individuals remain off of therapy with undetectable viral loads. So in each of these cases, the donor cells for the stem cell transplant had mutations. This mutation is called the Delta 32 mutation. And CCR5 is a receptor, a cell surface receptor. It's a co-receptor that HIV uses to enter and to infect a cell. And so this mutation confers resistance to strains of HIV that use this CCR5 receptor and confers natural resistance to HIV infection by some of these CCR5 tropic strains. So when these individuals received the stem cell transplant and their immune systems were repopulated with the HIV-resistant donor cells, it's thought that this is what led to the cure. There are also some other theories in the field about what led to cure in these cases. The first case of eradicating cure by stem cell transplant was the Berlin patient. His name was Timothy Ray Brown. Now, again, we have perhaps 9 other cases. This is what people may have heard in the media when cases of cure are reported. But turning to viral control off of therapy, or what used to be referred to as functional cure, there are now a few cases of people who are controlling HIV viral loads to very low levels after participating in a curative intervention clinical trial. So these cases come from several different trials testing different strategies. Most of them test what's called broadly neutralizing antibodies. And the individuals who control viral loads off of therapy are called post-intervention controllers. So it's important to note, again, that in each of these trials, only one or a few participants controlled HIV for a period of time after the trial, and the majority of participants in these trials had to go back onto antiretroviral therapy. One example is a participant from a trial called eClear, conducted in Denmark, and this participant has been controlling HIV off of therapy for about 7 years now. So this individual still has a measurable HIV reservoir, which has been stable in size over time. But there are characteristics of this HIV reservoir that have changed over time that are suggesting very strong pressure by the immune system. So it appears this person's immune system, through multiple effector mechanisms, is maintaining control of HIV viral loads off of therapy through very strong immune pressures.
Okay, it makes sense now. And also, like, someone, I think a few weeks ago, sent me a message on TikTok, and I was like, yeah, I've been living with HIV for some time, but I haven't started treatment yet. I've just so scared, but I've been just going for my checkups and my blood tests and everything with my doctor, just controlling, well, not controlling, but making sure that the levels, my CD4 and viral loads are at good levels. So it makes sense now why he hasn't started yet and he hasn't fallen because of that. And also when you're giving examples of some of the patients that have been cured of HIV, I met 3 of them last year at the AIDS 24 when we're in Germany. So those of you who want to see them, you can just go and check out my TikTok. I think I'll just pin it for this episode so that you see them, and it makes sense. And now when we're talking about TikTok also, I post a lot of my advocacy work on TikTok. And last year, around this time, I posted about stem cells. And you mentioned stem cells when you were now talking about eradication cure. They were cured because of the stem cell transplants. And now one of the a lot of the comments that I get on my TikTok is that, yes, stem cells, especially those in, I think in South Africa and Kenya's side, East Africa and then in West Africa, I get a lot of comments of people claiming that they're selling stem cells or you can buy stem cells. Some of them are showing some packages to say that this is the stem cell tablets or you can drink this. This is stem cell. They package them in such a way that people are sort of like convinced that this is a stem cell. I'm going to drink this stem cell tablet and I'm going to be cured of HIV. So please explain what a stem cell is and can you buy stem cells and just drink and then you're cured of HIV?
Yeah, so unfortunately you cannot. So consuming stem cells or consuming whatever this product is will not result in a stem cell transplant. It won't result in a viable transplant of those cells. So unfortunately, it is not a cure, what's being marketed. So an actual bone marrow stem cell transplant is a very intensive medical procedure. It actually requires ablating your own immune system. And so it used to require actually total body irradiation. Now there are chemotherapies that are used to first sort of remove or eliminate your own immune system. It then requires a transplant with the donor cells, which is an infusion procedure. And then it takes time for your immune system to repopulate with the donor cells. So it's a very intense medical procedure. There's actually, unfortunately, it's not safe enough to use in most people living with HIV. There's a high morbidity rate, which means it has a lot of side effects. And there's still a high mortality rate. So I believe currently that it's still around 20%, meaning 20% of people who get this procedure do not survive. And so it is not safe enough to use in most people living with HIV. It's a highly medicalized intervention. It's also not scalable. So this concept of scalability ties into affordability. It's a very expensive procedure. So a stem cell transplant, at least in the United States, can cost well over $1 million US. And so it's not safe. It's not scalable. But you also can't consume the stem cells and result in a transplant and a cure of HIV, unfortunately.
Okay, no, thank you for just clarifying that because I get a lot of those comments and I will also just share with them so that they see that you can't just say that you're going to buy stem cells and drink them and those are not stem cells. Those are just people like you saying those are stem cells. So yeah, thank you for that clarification. And now I want to know now between the functional cure and sterilizing cure, which one do you think is more achievable in the near future?
Yeah, so, as I mentioned, demonstrating that eradicating cure or sterilizing cure has been achieved is very difficult, essentially impossible. But based on current evidence, it does appear that viral control off of therapy is a more realistic and achievable goal. So when we are testing different cure strategies, we need to consider what's called the efficacy. So in how many people does the cure strategy result in control? We need to consider durability. So how long is the control maintained? Is it just a few weeks? Is it a few months? Or is it several years and more? And then I mentioned this concept of scalability. So we need to consider how scalable to different populations and different geographic regions is the strategy. And so all those considerations in place, it does appear that, functional cure, viral control off of therapy is hopefully going to be achievable and certainly a more realistic goal than a complete eradication. It is the case, though, there's a concept in the field called reduce and control. So it is the case that maybe a curative intervention does result in some reduction in what's called the size of the HIV reservoir and may result in elimination of some HIV-infected cells, thus allowing the immune system to better control those remaining infected cells off of therapy. So I don't think the two are unrelated, but I think this concept of viral control off of therapy is more realistic and more achievable. And we're starting to see what I think are signals that viral control is possible.
Now, when we're talking about some of the latest HIV cure, and recently I think, yes, recently in March, we did hear during CROI some of the most exciting HIV cure clinical trials that were being conducted, and some of the results. And one of those was the fresh cohort that took place in South Africa. And it's been trending lately in Southern Africa, especially on social media, like on your Twitter, TikTok, and Instagram, where now people are now saying that there is finally an HIV cure in South Africa. We are free of HIV. Can you please just explain to us what the fresh study is, and then also where does it fall between these two types of cure that we're discussing today?
Yeah, so this the study of the fresh cohort is unique. Here, the participants in the trial were young women in South Africa who were diagnosed and started on antiretroviral therapy very, very early in the course of HIV. Most people are not diagnosed that early, so they were diagnosed and started treatment within the first few days of HIV after acquiring HIV. Most people do not get diagnosed that early and start treatment that early. The reason that is important is because we know now that if you start therapy very, very early, it results in what's called a smaller HIV reservoir and also a less genetically diverse HIV reservoir. So HIV doesn't have a lot of time to replicate. Thus, the diversity of the reservoir is very limited. And we think then it may be easier to control the virus off of therapy in those cases. And so in this clinical trial is a combination of broadly neutralizing antibodies with a toll-like receptor agonist, this immunomodulatory drug, and 20% of participants controlled the virus, I believe for up to a year. I have to look at the exact data. When it was reported, after interrupting antiretroviral medication. So they started HIV medications, they received these curative interventions, but 20% of participants controlled off of therapy. So I think it's important to highlight there that the other 80% of participants did not control the virus off of therapy. And so it's still unknown the exact mechanisms of how these 20% of people participants were controlling for up to a year off of therapy. And so that's still an active area of research and investigation to understand those mechanisms. But again, it's a very unique participant cohort. And so I would be very cautious in interpreting those results. They may not be generalizable or applicable to most people living with HIV who initiated therapy much later in the course of HIV.
Yes, and I think for the fresh cohort, I think they were off of therapy for, I think, 52 or 54 weeks.
Okay.
So, yeah. And then now when we're now talking about this, because people are so excited that, yes, we finally found a cure. It's not really a cure. It's just they're still on, testing clinical trials and seeing if it's something that can be done. And like you did say that the number is still very low, that 20% and then 80% had to go back on treatment. It shows that there's us that while we haven't really found a cure, or even though it's promising, yes, it's something that we can do more studies on, but it's not yet.
Absolutely. Yeah. And I think the other thing I would just highlight there is, again, these participants are still living with HIV, but their immune systems are controlling the virus, controlling viral replication. So I wouldn't call it evidence that a scalable curative intervention that's going to work in most people living with HIV has been developed. But it does highlight that these complicated cure-related clinical trials can be conducted in resource-limited settings. And so I think that's a very exciting advance that the trial showed.
Yes. And then now when we're talking about access and equity, when scientists, researchers are busy trying to find out or to research for this cure, why is it important to make sure that in the future of HIV cure, we have a cure that is accessible and affordable for all communities?
Yeah, absolutely. That's it's a very important consideration in the development of cure strategies, because ultimately we want a curative intervention or maybe a set of different curative interventions that can reach the majority of people living with HIV, including and especially people living with HIV in resource-limited settings, and especially people who don't have access to or don't take antiretroviral medications. So I think if there's anything that this current crisis in the United States with PEPFAR illustrates, it's just we must intensify our efforts to advance a cure or multiple different types of cures that are accessible to all people living with HIV. And I also think it's important to highlight that one strategy that works in a specific population may not work in every person living with HIV. And this could be due to differences in host genetics, viral differences, so difference in viral strains. So we must conduct inclusive research and inclusive HIV cure clinical trials, especially in high-burden geographic areas that can benefit the most from an eventual curative intervention, so that we understand what's going to work in those areas and then in those populations. So, this issue of accessibility, affordability, and scalability is central to the development of curative interventions. Right now, the field is really just trying to test what can work in any group of people and to expand our knowledge from there. So the field is still very early. Cure trials are still very small, early phase trials, mostly for safety, very little around efficacy. And so we're still just to test what can work in any person living with HIV and learn from that and build from there. But ultimately, we need an intervention or, again, multiple different types of interventions that are going to work and cover the entire population of people living with HIV.
All right, now that you were talking about the fresh cohort and explaining how it worked, then what role does the immune system play when it comes to now achieving this functional cure or the viral control of therapy?
Yeah, so many of the cure strategies currently being pursued in clinical testing and also preclinical investigation involve harnessing different parts of the immune system, such as what are called cytotoxic T cells, also B cells and antibodies, cells, and also components of the innate immune response. And it's currently unclear what combination of immune responses is needed to achieve viral control, and thus how to target and improve those specific responses. But progress is being made on this front and in studying all of these different aspects of the immune system. So, for instance, in the eClear participant that I mentioned, who's been controlling for several years now, it appears that a combination of what are called potent cytotoxic T cell responses, these are very strong T cell responses targeting HIV, as well as potent autologous neutralizing antibodies. So these are the participants' own antibodies against the virus that are able to neutralize cell-free virus. It appears that a combination of these responses are helping that individual. We still have a lot of work to do to figure out how to harness the immune system to achieve cure. But it is the focus of a lot of different current cure-related clinical trials and a lot of basic science investigation and preclinical investigation in animal models.
Okay, now I'm going to put you on the spot that if you now had to choose between sterilizing cure, so the eradication cure versus functional cure, viral control of therapy, which one are you going to pick?
Well, I'm not sure that I can answer this question directly because it's hypothetical. Yeah. I do think it's important to point out that bone marrow stem cell transplant, which has resulted in possibly 10 cures so far, mortality rate and risk for severe complications. So it is not a suitable procedure for the majority of people living with HIV. And also currently modern antiretroviral therapy is very safe. So curative interventions were also similarly need to be quite safe. It is thought maybe if there are some side effects of certain cure strategies that make it perhaps a little bit less safe than modern antiretroviral therapy, that may be acceptable, particularly for people who can't access antiretroviral therapy and who aren't on HIV medication. Perhaps a cure can benefit those individuals the most. So, the field does think that cure strategies, curative interventions need to be very, very safe. Procedures resulting in eradication are not going to be safe enough to use in the majority of people living with HIV.
Okay, all right. So safety is the most important. But of course, like you did say that there are pros and cons to each one of them. And, in Africa, I think one of the things or the reasons as to why people are so excited is that, they were not taking ARV for a year. So it means that there might be hope and all of that, which is, yes, it is quite exciting, but we also need to be careful in terms of not now encouraging people to be off of ARVs or to stop ARVs and saying that, no, I am undetectable, so I'll just control my HIV at home. So we're not saying that, no, please. And
right. We will talk about in the later episode groups of people called controllers. So it's a rare subset of individuals, maybe 1 to 5% of people living with HIV, who, if they stop taking HIV medications, they do control the virus to low levels for a period of time. We will talk about those individuals. We'll also talk about a group of individuals that the field calls elite controllers, who can control the virus. Their immune systems can control the virus without the need to start HIV medications, at least for a period of time. And so we'll talk about those rare individuals, but I would highlight, as you do here, that it's really a rare subset of people living with HIV, and most people really need to adhere to and take their HIV medications. So really need to encourage people to continue staying on their antiretroviral medication.
Okay, and then now we're heading towards the end, and to our listeners, we talked a little bit about VNABs and all the boosters and everything. So please don't worry, we are going to have an episode on that where we're going to explain more what these VNABs are, how do they work, the broadly neutralizing antibodies, and what role do they play in HIV cure. Now to just going towards the end of today's episode, Adam, I want to know now, we're talking community. When we started, we talked about person-centered language and the importance of putting our communities first, putting people first. How can then these communities help researchers develop and test these potential cures in an ethical and inclusive way? What role do they play?
Yeah, so this is a great question. You know that community participation in research is imperative and will be imperative for the eventual success of a cure. I think an important starting point for researchers is for researchers to engage community members by providing accessible, understandable information about the current state of HIV cure science, which I think your podcast is helping to do. I think it's important for us to educate on what types of therapeutic interventions we are testing in clinical trials, so that communities understand what we are asking of them. But we have to build, as researchers, long-term relationships with communities and with community stakeholders. So I think one positive example I can point to is the Martin Delaney Collaboratory Program in the U.S. Again, this flagship program for HIV cure science. It's funded by the U.S. National Institutes of Health. And so here, each collaboratory, which the group that I help manage is one of those collaboratories, has a dedicated community advisory board and community engagement component, which we have worked with for several years. And there are 10 of these groups. And so I think this MDC program has really helped with community engagement. But broader than that, we have to build and maintain long-term relationships with community stakeholders and different communities impacted by HIV, and continue to provide information understandable, accessible information about cure science. I think then we also have to provide accurate information about different cure strategies that we're testing in clinical trials. So we have to have accessible participant consent forms. And so a lot of onus on researchers. And I do believe that as a community of researchers, we need to do more to promote accessible information on HIV cure science. But it has to be a two-way interaction, and it has to be an ongoing interaction and an ongoing long-term relationship.
All right. Thank you. And also, when you're talking about, just communities, we do see organizations like the IAS, the International AIDS Society, and AVA that are creating these programs for people, young leaders across the world who are interested in HIV cure or in even HIV prevention clinical trials to apply for these programs. HIV Vaccine Academy, the IAS HIV Cure Academy that I was part of, and that's where I met you, so that we were able to be empowered to learn and also just see what we can do in our communities and how is it that we can do it in such a way that people are able to understand and also learn that, oh, this is what's happening in clinical trials, or these are the clinical trials that are happening in Botswana or in Africa, and how do we engage or how do we become a part of it. So I also appreciate these programs that are there, and also just you being a part of it. It's really commendable. And just to close it off, I am going to put you on the hot seat again on the spot to say that if let's just be delusional here and say that we're gonna forget about all the challenges that are there in HIV cure. If you are now designing a perfect HIV cure, what's the one feature that it must have?
Just a few features. I think I'll reiterate the importance of scalability. So a cure that costs $1 million per person, right, is not going to be scalable. But I think when we're talking about cure strategies, even cure strategies that may right now cost $1 million per person, such as some strategies that use engineered T cells, like what are called CAR T cells, chimeric antigen receptor T cells. That's just one example. I think it's possible that technological advances in the future can bring down the cost of current strategies that are being pursued. So we must keep that in mind as well as we're testing different cure strategies preclinically and in clinical trials. And so, yeah, I think scalability is an incredibly important consideration that we must make as we're designing these curative intervention and as they're going into the clinic. Yes, well, again, I thank you very much, Sagabo, for having me. It's a real pleasure and honor to be here. And I also want to say to listeners that I'm very happy to answer questions outside of this podcast and happy to provide my contact information as well.
Yes, thank you for making the time. And if you are listening and you have questions, please just you can comment, or if you want to send it privately, you can send it to me. And I'll also share on my social media Adam's email, like on LinkedIn. If you want to contact him and ask him to understand some of the things, you can do that. I'll share that on my LinkedIn on a post. And then, yes, let's continue learning and growing, and also just being empowered and advocating in our different countries for an HIV cure during these times of funding cuts and just we don't know what tomorrow holds. Thank you so much, Adam.
Absolutely. Thank you. Yeah, we have to work and advocate together.
All right. Thank you so much for listening, and I'll join you on the next episode. Bye.